Now, cell division is a very fundamental process to any cell. And more importantly, the regulation of the cell division is very important, because the division of the cells should be in appropriate amount, no more and no less. Now, when this cell division and its regulation is off the hook, and the cells are dividing, and the cells divide abruptly, forming lumps of cells. This condition is called cancer. For example, if this is your skin or epidermal, the outer layer of the epidermis, you have the cells over here, and the regulation is lost for these cells, and these cells starts to divide abruptly and rapidly. So you'll have various cells, growing in an uncontrolled manner. And finally, these cells, as they divide and form new cells, there would be a lump on your skin. This lump, we call it this lump to be the onset of cancer, and this lump, clinically, we call it tumor. So that is how cancer is actually mediated. That is how cancer starts. Now, for this regulation part, there are two kinds of genes that actually play a very important role. One of the type of genes, they promote cell growth. Normally, they promote cell growth. So when they're functioning in a normal manner, they're promoting the cell growth. We call it proto-oncogene. And there is another type of cell which represses the cell growth. We call it the tumor suppressor genes, or TSG, tumor suppressor genes. Now, both these type of genes, they have different roles to play. When I'm saying that the proto-oncogenes are promoting the cell growth, that is, they're positively regulating the cell division. When I'm saying that the TSGs, or the tumor suppressor genes, are repressing the cell growth, they are negatively regulating the cell division. What happens is that when the cell is moving in the cell division cycle, there are certain checkpoints in the cell division cycle, and these checkpoints has to be crossed. The cell need to cross this checkpoint in order to divide. When certain proteins, they sense that the cell is having any kind of damage, they try to repair the system first before the cell can move into the next phase. For example, let's say the DNA is, or the cell is exposed to UV, and this region of a particular gene or this region of the DNA is damaged because it had received UV radiation, and this part is damaged. Now, what certain proteins would try to do, they would try to repair. They would sense this damage, and they would recruit other proteins that would try to repair this DNA damage before the cell can move on into the cell cycle. So this is the work of the tumor suppressor genes. They won't let the cell proliferate if any condition in the cell is not favorable. Now, proto-oncogenes, what they do, they actually promote the cell growth. They send signals to the cell division machinery that the cell is ready for the cell division. Now, in cancer situation, what happens? These proto-oncogenes are mutated, and they have something called gain of function. So these are mutated and they have something called gain of function. Now, what is gain of function? The function that was already there in the proto-oncogenes, that is to promote cell growth, the function is enhanced in a certain way. Enhanced function. Now, as the function is enhanced, that means these proto-oncogenes are producing huge amount of the proteins that was being encoded by the proto-oncogenes. Now, when these proteins are being encoded in a huge number, they are abruptly causing a positive regulation towards the cell cycle. They are promoting the cell growth abruptly, and the cell division is going wayward. Now, for the tumor suppressor genes, they have something called, after mutation, they have something called the loss of function. Now, loss of function, what it does, it declines the function of the tumor suppressor genes, or it represses the function. So the tumor suppressor gene, normally what it was doing, it was repressing the cell growth. So this was its normal function, repressing the cell growth. Now, how it was repressing, it was not allowing, it was sensing the damage, and it was not allowing the cell to move into the different phases of the cell cycle, or it was not allowing the transition through different phases of the cell cycle. Now, once it is mutated, this function is not there. So the TSGs cannot sense the damage, but the proto-oncogenes, these are active. These sort of promote the cell growth, and the cell divides in a normal way. Or when the damage is there, the cell divides with the damage. So if there is a DNA damage, if the tumor suppressor gene is mutated, then this mutation would actually not sense the DNA damage. Since the TSG is mutated, it won't sense the DNA damage, and the cell would sort of proliferate, and it would have that DNA damage within the cells that it is making, and that causes the onset of cancer, because the cells are growing in an uncontrolled manner, and the damages in different areas of the cells are not maintained by the different genes that should be there. So the abrupt division of the cell finally causes this formation of tumors. And when the tumor grows, as the cells are growing, the number of cells is increasing, so the metabolic activity of the cells are also increasing, and as a result, they need a lot of nutrients that needs to be supplied for these cells. But the blood vessels that was there was not sufficient for these new cells. So what the tumor does, it starts making new blood vessels. so that oxygen and nutrients can be supplied to them through these new blood vessels. And this process of forming new blood vessels by the tumor cells, we call it angiogenesis. Angiogenesis. So as new blood vessels are formed, the new tumor cells that are growing abruptly, they would receive nutrition and grow farther. So that was a little bit of introduction to cancer. So we talked about the proto-oncogene, whose normal function was to promote cell growth, and when they're having a gain of function, their function is enhanced. That is why there is abrupt cell division, because they are secreting more proteins than they should have been secreting. So more number of proto-oncogenes, that means the cell division is enhanced, and abrupt division causing the formation of tumors. On the other hand, the tumor suppressor genes, they are repressing the cell growth, and they actually senses any damage to the different compartments of the cell. They send out signals to different components of the cell so that the damage can be repaired, and then the cell can move through the cell cycle. But when these TSGs are having a loss of function, this property is lost, and since they're having a loss of function, the TSG's function is actually repressed, and that causes the cell to move on with the damage, and that causes the formation of tumor and abrupt cell division. Finally, when the tumor is growing, they would require new blood vessels that would supply them with oxygen, nutrients, and other stuff. So they are forming these new blood vessels, and we call this process the angiogenesis.